Hello, and welcome back to another episode of Teach Me in 10. In this series, we challenge scientists to break down their research into 10 minutes or less, making complex topics accessible and engaging for everyone. Today's episode is brought to you by Thermo Fisher Scientific, whose mission is to enable their customers to make the world healthier, cleaner, and safer. Whether it's accelerating life sciences research, solving complex analytical challenges, improving lab productivity, or developing life-changing therapies, Thermo Fisher is dedicated to empowering breakthroughs. To learn more about their innovative products, check out the provided resources. I'm Brie Foster, science writer at Technology Networks, and I'm delighted to introduce today's guest, Brandon Bills, product manager at Thermo Fisher Scientific. Brandon earned his PhD in analytical chemistry in 2019 from Purdue University in Indianapolis. He joined Thermo Fisher Scientific in 2020 as a product specialist focused on small molecule applications on Orbitrap Tribrid mass spectrometers. Since then, he has been promoted to product manager of the Orbitrap IQX Tribrid mass spectrometer, where he focuses on helping customers generate high-quality data from complex samples. In today's session, Brandon will walk us through real-time live researching, an innovative feature on Orbitrap Tribrid's instruments. This technology enables users to design the methods that match fragmentation data to a library in real time. Based on these matches, the system can decide whether to proceed with secondary characterization scans or move on to analyze other precursors.
So today I want to talk to you and to everyone else about how you can use library data that you've already collected to guide an untargeted analysis towards more relevant results. Specifically, I'm going to talk about forward and reverse cosine scoring in real time to trigger additional experiments when you know there's something you want to know more about. In this case, it'll be a part of a collaboration where we're looking at inbuilt compounds. But I'll get more into that later. First off, my name is Brandon Bills. I am a product manager at Thermo Fisher Scientific, based here in San Jose, California. And I've got my PhD in analytical chemistry back in 2019 from IUPUI. Real quick, all this work was done as part of a collaboration with Michael Christopher and Tim Garrett at the University of Florida. All of the data you see was collected on Thermo Fisher Scientific instrumentation and software. So we'll start off with what is real-time library search, or RTLS, on the Thermo Scientific Orbitrap IQX Tribrid mass spectrometer. We'll talk about what specific problem are we trying to solve, and the different types of experiments you can run, as well as the scoring options you have available. Then we'll go into a real example where we're looking for indole compounds in a complex matrix. And for that, we're specifically looking at what are indole compounds, how real-time library search changed the acquired data, and how that in turn changed the final results. So the big question to start off with is, what is real-time library search? So this is a filter that, if you know our drag and drop method editor, goes in after your MS2. This MS2 will take in your experimental spectra and compare it to a library in real time, compare it against scores, and say, yay or nay, run the experiments that are beneath this node. You give it a library and some score thresholds. Typically, these are empirically determined based on what you know is around the level of confidence you need. And then the instrument knows, okay, I only need to spend time running these further experiments if it's actually going to help. Put that in perspective. Without real-time library search, your typical brute force experiment, you'll collect MS1, you'll choose a target to collect a data-dependent MS2 on, and then, regardless of how important it is, it will collect everything else that's in your method. Like in this case, UBPD and 3MS to the third. You get sort of a confidence that you are collecting data on everything, but it's very inefficient if there's things you don't want to study. So let's look at something that's a little bit more efficient in that regard. With real-time library search, after the MS2, there is a bit of a break where it will check that library with your experimental fragments and see if there are matches. If there's enough matched compounds to cross your threshold, then you know it could be interesting, that it's worth time spending instrument resources collecting more data. If it's a complete mismatch, it's likely not related to what you're interested in. So it's better to spend that time moving on to something else that might be interesting. And that's really the core of what real-time library search aims to do, is just make your untargeted experiments a bit more intuitive about what the data it's spending its time collecting. So why would you need to do this? Well, we all know that not every compound will be interesting to your study, or maybe even not need additional information to fully characterize it. Your library represents knowledge you already have about what is important to your work. Real-time library search uses that to make your final results more concise and more relevant to what you want to know. We've got a couple of experiments you can run. The first one I'll talk about is an identity type experiment. In this one, you match the precursor M over Z from the MS1 and the fragments. This is where you're trying to sort of confirm what you have is exactly what matches in your library. Alternatively, if you just want to find things that are similar, as in they have shared structural characteristics, you don't really care if you match the precursor M over Z. Instead, we skip that and go straight to comparing MS2 fragments. And the goal here is to find things that share fragments, because that's usually a clue that something is structurally similar between your experiment and your library. And that's really what we're going to focus more on today. Let's talk about the different scoring options you have available when you're doing real-time library searching. So when you give it a spectra to process against the library, it's going to try to generate a score based on the similarity. There's a couple of ways of doing it. There's a lot of nuances and math behind this, so I'm just going to focus on what's important for our talk today. I've got an example of an experimental and a library spectra, and you can see there's some matching peaks and there's some mismatching peaks. In forward scoring, you're checking the library for your experimental peaks. Matches will make your score higher, while missing experimental peaks will push the score lower. And we'll talk about why that's important for your type of experiment later. In reverse scoring, you're just looking to see, okay, I've got these library fragments that are present in the experimental. This means that forward scoring is going to be better suited for a confirmation, like in an identity type experiment, because you're trying to confirm that all of your experimental fragments are present in the library, or as many of them as possible. Whereas reverse scoring is better suited for similarity type scoring, because you are looking to see, are these fragments that tell me something is similar to my library present in the experimental? So how does all of this help overall? Those of you who have used a tribrid before know that you can, with iterative analysis, study everything via sample. And you can collect additional information like MS to the N or alternate fragmentation to help characterize those unknowns. But you also know this leads to a huge data set. It takes time to study everything. Real-time library search helps focus the instrument on what is going to be most valuable to you. For example, an identity type experiment would say, don't waste time studying when I've already got it annotated. It's a perfect match to the library. Or a similarity type experiment would say, there's a subset of chemistry that I really need to know more about. When you see this, run these experiments for me. That way, when you get to the end, you've got as much data that you do need while minimizing the data that you don't. This really brings us to our collaboration with Michael Christopher and Tim Garrett. They came to me wanting to study indole compounds in plant extract. Started off by providing me a library from indole-containing standards. There's some examples here. Fun fact, the one at the bottom there has a much shorter and more common name. It's also known as melatonin. So these are biologically relevant compounds that they wanted to know more about, especially if there were any unknowns that they'd missed previously. We curated that library to remove anything that was simple, so only one or two peaks, because those can cause false positives. And then we analyzed plant extract with and without real-time library search in a similarity type experiment with both forward and reverse scoring to see how it impacted the results. First off, let's talk about the distribution of scores. So when we ran this experiment with forward scoring, we had around 6.6 thousand MS2 collected. Out of those, 282 had cosine scores above the threshold we set of 20, 20 being where we saw a drop off in any amount of similarity. And 193 of those fell between 20 and 30. We really only saw high scores with this if it was already a pretty close match to the library. You can see 55 and higher, that's going to be the things that were pretty much the library entries already. That's because any additional experimental fragments relative to the library push that score lower. And that means that if you have something that's got some similarity, but it might have an additional functional group, you might just miss it because forward scoring doesn't account for that. Reverse scoring, however, out of 5.3 thousand MS2 collected, 498 of those had scores above 20. And they were much more distributed between 20 and 90. That's because they only depend on whether or not the library fragments are present. Again, the big take home here is that if you're doing a similarity type experiment, forward scoring, which is better for an identity match, is going to push those scores lower because of those additional functional groups causing fragments that it's not accounting for. Whereas reverse just looks for your library fragments. So this changes how the instrument spends its time. That's the big thing we're doing here. And let's break this down by the different scan types that were collected. On the left here, we have your MS1 and MS2. Let's call those our scans to find compounds. This is where we initially see if something is worth further investigation. On the right, we have the additional information. These are the experiments you set up to make sure that you have everything you need to characterize any new unknowns when they crop up, so that you don't have to go back and reanalyze a sample. Without real-time library search, we see the results of brute force. For every MS2 that's collected, we see we've got a similar number of UVPD scans collected, as well as 3 times that number of MS3. That's how we set up the experiment. It's perfectly logical why those results look like that. With real-time library search, however, we see we reclaim a lot of that time we spent collecting additional scans on things that weren't related to the library. So we find a lot more compounds, but importantly, we still collect that additional data on the compounds of interest because we told it ahead of time, I want to study things that are similar to what's in my library. So how does that change your final results? The big question. So we processed all the data in Thermo Scientific Compound Discoverer software. We were looking at compounds that had MS2 collected and had one or more known indole class fragments. Now, when I say indole class fragments, I mean if you look through the library of indole compounds, these are the fragments that were found over and over again throughout that library. Then we looked to see if it actually triggered additional data collected. And that told us, did we find the most of what we were interested in? So we can see that just looking at the compounds that triggered MS2, no real-time library search triggered on the fewest number of these potential indole compounds. That makes sense. It's brute force. It's spending most of its time collecting the additional data. We see forward scoring found the most of these potential indole compounds for MS2. But that's because it refused to run the additional experiments on anything that didn't match well to the library. And if we look at what had additional data collected, we can see that's actually reverse scoring that found the most of these potential indole compounds and triggered the additional experiments on them. In fact, we see that about 49% more of these compounds relative to no RTLS at all had that additional data collected. So it really gives more concise and relevant results in the end using the library data you already have. So let's wrap this up. We use real-time library search on the Orbitrap IQX to try to really refine our results to what we want to know. Real-time library search focuses the instrument on what's most relevant based on the library and the score thresholds you give. And in this particular case, reverse scoring is going to be better suited for similarity matching than forward scoring. Though I will say, if you are trying to match something identically in the library, forward scoring is going to be the better tool. So I'd like to acknowledge all my Thermo Fisher Scientific colleagues, as well as all our collaborators at University of Florida, the Department of Chemistry, Michael Christopher and Timothy Garrett especially. And I'd like to thank you all for your attention today.
As always, thank you all for tuning in to Teach Me in 10. Don't forget to subscribe so you don't miss any more quick science bites. See you next time.